Collective cell migration is an important process in both normal development and tumor metastasis. The cadherin family of intercellular adhesion molecules promotes collective cell migration, and Cécile Gauthier-Rouvière from the CRBM Institute in Montpellier was interested in the role of one particular family member, P-cadherin.
P-cadherin is overexpressed in several invasive tumors, like breast tumors, but also pancreatic tumors. And it was known that P-cadherin expression is a marker of poor prognosis and is associated with an increase in invasion and migration. But how it occurs was unknown. So this is why we started really to study its role.
To investigate the role of P-cadherin, Gauthier-Rouvière and colleagues, led by graduate student Cédric Plutoni, expressed the adhesion protein in C2C12 cells, a myoblast cell line that doesn't usually undergo collective cell migration. The researchers then performed a 2D migration assay, in which cells move into a free space created by the removal of a physical barrier.
So when we express P-cadherin in C2C12 cells, we first observe that the cells move faster than the control cells, but more importantly, they have a different migration behavior. They migrate in a more efficient and collective way, all together in the same direction.
Quantitative analyses confirm that, although P-cadherin has no effect on the motility of isolated C2C12 cells, it promoted the collective migration of confluent cell layers by increasing the velocity, directionality, and persistence of cell movements. Expressing E or R cadherin, in contrast, had no such effect on the collective migration of C2C12 cells.
Also, by monitoring the position of the centrosome and of the Golgi apparatus relative to that of the nucleus, we observed that P-cadherin expression increased the polarity of the cells. And looking at the organization of the cell layer using actin staining, we observe also that the typical protrusion that is formed at the front of the migrating cells is highly polarized in P-cadherin expressing cells compared to the other cell line.
Plutoni et al. Also examined the organization and dynamics of the cell matrix adhesions formed in P-cadherin expressing cells.
And we observed that P-cadherin expression specifically oriented the focal adhesion in the migration direction, both at the cell front but also inside the cell layer. This proved that cadherin, which is expressed and located at cell-cell junction, is able to induce a response at the distal cell matrix adhesion site.
Indeed, cadherins can affect the mechanical forces transmitted at both cell-cell and cell matrix adhesions, both of which are important during collective cell migration. Cells migrate by exerting traction forces on their underlying substrate, while intercellular forces help guide the cells in the right direction. Plutoni et al. Therefore teamed up with Javi Tripau and colleagues at the University of Barcelona to examine how these forces were affected by P-cadherin expression.
And what we observe is that P-cadherin expression not only increases the strength, but also the anisotropy of the intercellular forces. P-cadherin expression is associated with an orientation of the maximal stress parallel to the migration direction. And this was not observed after E or cadherin expression. We also measure the traction forces, which are the force that the cell exerts on the substrate. And we observe that P-cadherin, again, is associated with a specific increase in the traction forces and also an increase of the orientation of the traction forces in the migration direction.
Thus, P-cadherin drives collective cell migration by polarizing cells and orienting mechanical forces in the direction of migration. Plutoni et al. Therefore examined whether P-cadherin regulates any of the Rho GTPases that control cell polarity in the actin cytoskeleton, and found that P-cadherin expression specifically activated the GTPase Cdc42.
Then we wanted to identify the regulator of Cdc42 activation downstream P-cadherin, and to do so we performed a proteomic analysis of the molecules that were immunoprecipitated with P-cadherin, and we identified beta-peaks, a guanine nucleotide exchange factors involved in Cdc42 and RAC1 activation. And we show that Cdc42 activation by P-cadherin is abolished in cell knockdown for beta-peaks.
Furthermore, knocking down beta-peaks or Cdc42 inhibited P-cadherin-induced collective cell migration, reducing, for example, cell persistence and polarization, and impairing the generation and orientation of both intercellular and cell substrate traction forces.
So in conclusion, we demonstrated that P-cadherin, which is a protein that is upregulated in invasive carcinoma and sarcoma, promotes collective cell migration through an increase in strength and anisotropy of mechanical forces. And also we identified the P-cadherin beta-peak Cdc42 axis as involved in the induction of these mechanical forces and generation of collective cell migration.
Beta-peaks can associate with other members of the cadherin family, but only P-cadherin was able to recruit this protein to cell-cell junctions, and this recruitment only occurred during collective cell migration. Gauthier-Rouvière and colleagues now want to investigate how beta-peaks recruitment is regulated and to determine how P-cadherin is able to influence traction forces exerted at cell matrix adhesions. In the meantime, however, you can learn more about how P-cadherin promotes collective cell migration via a Cdc42-mediated increase in mechanical forces in the paper by Plutoni et al., published in the January 18, 2016 issue of the Journal of Cell Biology.
You